The compound should have killed him in 11 seconds. Dr. Tessara Vaughan had calculated this figure herself, 3 times, using 3 independent pharmacokinetic models calibrated against 400 years of toxicological data spanning 912 known species. The Presidium's own certified databases confirmed her number. 11 seconds from introduction to full systemic collapse. The neural inhibitor designated KR7 was not ambiguous in its effects. It did not leave room for partial outcomes. It shut down the autonomic nervous system in a cascading sequence: brainstem first, then cardiac rhythm, then respiratory drive, with the mechanical precision of a circuit being switched off at the breaker. Every species in the conclave's biological registry responded identically. 11 seconds, unconsciousness at 4, cardiac arrest at 7, brain death at 11. The human lasted 41 minutes, then he asked for water. Tessara stood at the witness lectern in the lower well of the Presidium Judiciary Chamber and tried to keep her hands from shaking. The chamber was vast, a tiered amphitheater of polished basalt and silver-veined composite, curved inward like the inside of a shell, lit from above by diffused luminescent panels that cast everything in flat, shadowless institutional light. 9 tribunal judges sat in the upper tier behind a continuous curved bench of dark stone, their robes the deep indigo of the Presidium Judiciary, their faces arranged in various configurations of professional neutrality. Behind them in the observation gallery, perhaps 200 spectators, academics, military liaisons, intelligence analysts, journalists from 6 systems, sat in ascending rows, their silence heavy and complete. Below the judges, on the chamber floor, 2 tables faced each other across a gap of perhaps 4 meters. The prosecution occupied the left table, Senior Advocate Dremes Toreth and his staff of 3, their datapads arranged with surgical neatness. The defense occupied the right, a single advocate, a young Veshan woman named Kel Sareth, whose crest had not settled once in the 4 hours since the hearing began. And between the tables, in a transparent walled witness enclosure that was standard for biological testimony, meant to contain airborne agents in pharmacological cases, sat the subject. The human. His name was Cole Brannock, 39 years old, former emergency physician from a Terran colony called Murren's Landing. He sat in the enclosure's single chair with his legs crossed at the ankle and his hands resting on his knees, watching Tessara with an expression she had spent 4 days trying to categorize and had finally settled on calling patient. Not hostile, not anxious, not defiant. Patient the way someone waits for a train they know is coming. Dr. Vaughan, Senior Advocate Toreth said from the prosecution table, please describe for the tribunal the events of the 14th of Tessek, beginning with the administration of KR7 to the subject. Tessara adjusted her spectral lenses, an old habit, a comfort gesture, and opened her testimony file on the lectern's embedded display. She had reviewed these notes 900 times. She knew every data point by rote, but the lectern gave her something to look at that was not Cole Brannock's calm, unhurried face. On the 14th of Tessek, at 600 hours standard, the Biological Assessment Division of the Cathara Research Institute initiated a scheduled pharmacological evaluation of the human subject designated BRN 4412, she said. I was the lead researcher. My team consisted of 11 specialists across 4 disciplines: neuropharmacology, xenobiochemistry, cardiac physiology, and metabolic tracking. The evaluation was part of a broader classification study commissioned by the Conclave Bureau of Species Assessment. Its purpose was to establish baseline responses to standard conclave chemical agents in Terran biology. Standard agents, Toreth repeated, meaning the 64 compounds in the conclave's standard pharmacological reference. Every species entering the galactic community undergoes this battery. The compounds include sedatives, analgesics, neural inhibitors, vasoconstrictors, immune suppressants, and metabolic disruptors. The results establish a species' chemical vulnerability profile, their tolerances, their thresholds, their lethal dosages. This data is used for medical treatment protocols, containment procedures, and threat classification indexing. And KR7 is one of these 64 compounds. KR7 is compound number 31, a neural inhibitor. It is the single most reliable unconsciousness-inducing agent in the conclave pharmacopoeia. It has been tested on 912 species. In every case, onset of unconsciousness occurs within 4 seconds of intravenous administration. Full neural shutdown within 11 seconds. There has never been an exception. She paused, the chamber waited. Until the 14th of Tesek. Toreth leaned forward. Walked the tribunal through the administration. Tessara touched her display and a holographic rendering appeared above the lectern. The Cathara Institute's Evaluation Suite 3, reproduced in precise three-dimensional detail. The suite was a clinical space, white-walled and bright, with a central biomonitoring platform surrounded by 12 workstations. In the center of the rendering, a figure sat on the platform, the human, rendered in anatomical transparency, his circulatory and nervous systems traced in blue and gold. Mr. Brannick was seated on the primary monitoring platform at 612, Tessara said. Full-spectrum biosensors were active. Heart rate, neural activity, blood chemistry, metabolic rate, respiratory function, immune markers, all monitored in real time across 12 stations. My colleague Dr. Pellari administered KR7 intravenously via a standard infusion port in the subject's left forearm. Dosage was calculated at the standard interspecies rate, 0.3 milligrams per kilogram of body mass. For Mr. Brannick's weight of 84 kilograms, this equated to 25.2 milligrams. A standard dose. The same dose that has rendered 912 other species unconscious within 4 seconds, without exception. What happened? Tessara enlarged the holographic rendering. The anatomical transparency of the human figure pulsed with data, glowing lines of metabolic activity, neural firing patterns, blood chemistry markers cascading in columns of numbers. At the moment of administration, 6:12 and 14 seconds, KR7 entered Mr. Brannick's bloodstream through the antecubital vein in his left arm. Our sensors tracked the compound as it moved through his circulatory system. Within 1.3 seconds, it reached his hepatic portal system. She highlighted the liver in the rendering. It glowed amber. At this point, in every other species, KR7 passes through the liver with negligible metabolic interaction. The liver does not recognize it as a threat. It is a synthetic compound engineered specifically to evade hepatic filtration, one of its design features. In 912 species, the liver processes less than 2% of KR7 on first pass. The remaining 98% continues to the brain, where it binds to autonomic regulatory receptors and initiates shutdown. What happened in Mr. Brannick's liver? Tessara looked at the rendering. She had stared at this data for weeks now, and it still produced in her a feeling she could only describe as vertigo, the cognitive sensation of watching a fundamental assumption dissolve. Mr. Brannick's liver processed 41% of the KR7 on first pass. The chamber shifted. A murmur passed through the observation gallery like a wave through water. One of the tribunal judges, the Veshan in the center of the bench, Judge Advocate Korath, made a note on his datapad with a motion that was perhaps slightly less steady than it had been a moment before. 41%, Torath said. On first pass, his hepatic enzymes, specifically a class of metabolic proteins that we have since designated CYPT7 through CYPT23, recognized the compound as foreign material and began breaking it down, not slowly, not partially. They dismantled it at the molecular level with an efficiency that exceeded any hepatic response in our database by a factor of 19. Factor of 19. The next most efficient hepatic response to KR7 in our records belongs to the Deadliest, a species whose liver processes approximately 4% on first pass. Mr. Brannock's processed 41%, and it accelerated. She advanced the rendering. The liver glowed brighter, amber shifting toward white. Data streams cascaded faster. By the time the remaining 59% of the compound reached Mr. Brannock's brain, approximately 2.1 seconds after administration, his liver had already upregulated enzyme production. The CYPT proteins were multiplying. Not through cellular division. The liver cells themselves were not replicating. The existing cells were increasing their individual enzyme output. Each hepatocyte was producing more metabolic machinery to process the foreign compound. That is not standard biological behavior, Judge Korath said from the bench. His voice was carefully neutral. Tessara looked up at him. No, Your Honor, it is not. In standard biology, enzyme upregulation in response to a foreign compound takes hours to days. The liver detects the compound, signals for increased enzyme production, and the cell's protein synthesis machinery responds gradually. In Mr. Brannock's case, the upregulation was measured in seconds. His liver quadrupled its CYPT enzyme output within 8 seconds of KR7 administration. By 20 seconds, the enzymatic activity had increased by a factor of 11. The compound was being destroyed faster than his circulatory system could deliver it to his brain. She paused and looked at the witness enclosure. Cole Brannock was watching her with what she had come to recognize as mild interest, the expression of someone listening to a moderately engaging lecture about a subject they already understood. What did the reduced compound concentration do to the neural effects? Korath asked. The 59% that reached his brain on first pass did bind to autonomic receptors. Mr. Brannock experienced a brief period of drowsiness. He reported afterward that he felt, and I am quoting from his post-evaluation interview, a little sleepy like after a big lunch. His heart rate decreased by 4 beats per minute. His eyelids lowered slightly. Then his liver caught up. Within 15 seconds of administration, the hepatic clearance rate exceeded the delivery rate. The compound was being metabolized faster than his blood could carry it to target tissues. His drowsiness faded. His heart rate returned to baseline. By 30 seconds, his blood concentration of KR7 had dropped below detectable levels. In 30 seconds. The compound that renders 912 species unconscious within 4 seconds was completely eliminated from his system in half a minute. He was fully alert, fully oriented. His neural activity showed no residual depression. He looked at Dr. Pellari, who was, at that moment, standing beside the monitoring platform with a syringe in one hand and her mouth open, and he said, "Was that it?" Another murmur from the gallery. Tessara waited for it to subside. "We administered a second dose immediately," she continued. "Double concentration, 50.4 milligrams. The response was identical but faster. His liver processed 63% on first pass. Blood clearance time was 19 seconds. He reported no drowsiness at all from the second administration. You increased the dose again. 3 more times over the next 40 minutes. Each time we doubled the concentration. By the third administration, 100.8 milligrams, 4 times the standard dose, Dr. Reth at our cardiac station began recalibrating his instruments. He assumed they were malfunctioning because the data showed Mr. Brannock's heart rate had not deviated by more than 2 beats per minute from his resting baseline across all 3 administrations. The instruments were functioning correctly. Mr. Brannock's cardiac system simply was not registering the compound as a meaningful event. She advanced the holographic rendering again. A new data overlay appeared, a cascading comparison chart showing blood clearance rates across the 5 administrations. each line steeper than the last, converging toward the time axis with the inevitability of a mathematical proof. By the fourth administration, 201 milligrams, 8 times the standard dose, something new happened. Dr. Pellari noted it first. Mr. Brannick's renal function had shifted. His kidneys had begun filtering metabolic byproducts from the hepatic breakdown of Kr7 at an accelerated rate, not the compound itself, the waste products. His liver was destroying Kr7 so efficiently, and so quickly that the volume of metabolic debris was exceeding normal renal processing capacity, so his kidneys upregulated to match. 2 organ systems, coordinating in real time, without any conscious direction from the organism. A coordinated systemic response, Judge Tessick said from the far end of the bench. A Kalori woman whose silver-scaled face had not changed expression once during the testimony. Yes, the liver leading, the kidneys supporting, and by the fifth administration, 403 milligrams, 16 times the standard dose, we observed a third system joining the response. His bone marrow had increased production of a specialized transport protein that facilitated the delivery of hepatic enzymes to peripheral tissue. His body was not merely processing the compound at the liver. It was distributing its countermeasures systemically, ensuring that any Kr7 molecules that bypassed hepatic filtration would be intercepted and neutralized in the tissues themselves. She paused. The chamber was very still. 3 organ systems, coordinating, accelerating, learning in 41 minutes. At this concentration in any other known species, the compound would cause not just unconsciousness but permanent neural tissue damage, irreversible. In Mr. Brannick, the hepatic clearance rate had accelerated to the point where his liver was destroying the compound in approximately 4 seconds. He experienced, by his own report, a slight tingling in my fingertips. Nothing else. And then he asked for water. He said the process was making him thirsty, which was the correct physiological response. The accelerated metabolic activity was consuming additional fluid resources. His body was telling him to hydrate because the increased enzymatic workload required it. Even his thirst response was calibrated to the defense his body was mounting. He asked if we had anything to eat as well, because he had skipped breakfast. Dr. Pellari handed him a water container. Her hands were shaking. His were not. Tessara advanced the rendering one final time. The holographic liver now blazed white, a dense cluster of metabolic activity that made the surrounding organ systems look dormant by comparison. This is the scan we took at minute 41, immediately after the 5th administration, she said. His liver had not merely adapted to Kr7. It had built an entire enzymatic architecture specifically designed to destroy it. The CYPT proteins we identified, 17 novel enzyme variants that do not exist in any other species in our database, had been synthesized, deployed, and optimized in under 41 minutes. His body treated one of the most sophisticated pharmacological agents in galactic medicine, the way a furnace treats paper. She looked at the tribunal. We did not proceed to compound number 32 that day. The chamber was quiet. Tessara could hear the soft hum of the environmental systems, the faint click of a stylus on a datapad somewhere in the gallery, the sound of her own breathing. Why not? Torith asked. Because I needed to understand what had just happened before I introduced another variable, and because Dr. Pellari resigned from the study that afternoon. She told me, privately, in the corridor outside the evaluation suite, that she had spent 11 years studying xenopharmacology and had never once encountered a result that made her want to leave the building. She left the building. Did you continue the study with your remaining team? I did. Over the following 9 days, we administered all 64 compounds in the conclave standard pharmacological reference to Mr. Brannick, 1 per session, with full monitoring. Before we discuss the full battery, Toreth said, I want to ask about specific compounds beyond Kr7. Compound number 8, vasotrine. A vasoconstrictor used in emergency hemorrhage control. What happened? Vasotrine restricts blood flow to extremities by triggering smooth muscle contraction in arterial walls. Standard onset is 12 seconds. In Mr. Brannock, the compound reached the arterial smooth muscle, began to trigger contraction, and was then intercepted by a counteragent his own body produced. His vascular endothelium, the lining of his blood vessels, released a vasodilatory compound that directly opposed the vasotrine. Within 9 seconds, the constriction was neutralized. By 15 seconds, his circulatory dynamics had returned to baseline. His blood vessels rejected the instruction. Compound number 44, the immune suppressant Deadliest. This one was particularly instructive. Deadliest suppresses immune function by inhibiting leukocyte activation. It is used in transplant medicine across 300 species. In Mr. Brannock's case, the compound entered his bloodstream and encountered his white blood cells as intended. His leukocytes recognized the suppressant, and instead of being inhibited, they phagocytized it. They consumed it. His immune cells treated the immune suppressant as an invader and destroyed it using the same mechanism they would use against a bacterium. The compound designed to shut down his immune system was eaten by his immune system. A sound came from the gallery, not a murmur this time, but a single, sharp intake of breath from someone who understood the implications. Tessara waited for it to pass. Compound number 57. The metabolic disruptor Nexethane. Nexethane interferes with cellular energy production. It blocks the electron transport chain in mitochondria, starving cells of their primary fuel source. It is the most sophisticated metabolic weapon in the standard battery. In Mr. Brannock, the compound reached his mitochondria and began its blocking mechanism. For approximately 4 seconds, his cellular energy output decreased by 11%. Then his mitochondria switched to an alternative metabolic pathway, a backup energy production system that his cells activated autonomously. His cellular output returned to normal within 8 seconds. By 12 seconds, his liver had neutralized the compound. But the important thing is the backup pathway. Why? Because it is not a known human metabolic pathway. We consulted the Terran biological databases after the session. Human medicine has documented certain alternative energy pathways, anaerobic glycolysis, ketone body utilization. But the pathway Mr. Brannock's cells activated does not appear in their own medical literature. His body used a metabolic mechanism that human scientists have not yet discovered in themselves. The vertigo was back. Tessara steadied herself against the lectern. And the results of the complete battery. Tessara felt the ground-shifting sensation that had become her constant companion over the past 3 weeks. She had published 47 papers in her career. She had contributed to 3 conclave species classification revisions. She had sat on the Cathara Institute's review board for 6 years. None of it had prepared her for what she had watched happen on that monitoring platform over 9 days. Of the 64 compounds, Mr. Brannock's body neutralized 61 within detectable parameters. Of those 61, 47 were eliminated in under 1 minute. 32 were eliminated in under 30 seconds. 9 were eliminated so rapidly that our instruments could not establish a peak blood concentration. The compound was being destroyed faster than we could measure its arrival. She paused, let the numbers land. The 3 compounds his body did not neutralize were analgesics, pain modulators. His liver processed them more slowly, and they produced their intended effect, mild pain relief. Mr. Brannock described the sensation as taking an aspirin. They wore off within 20 minutes. So his body destroyed every hostile compound, but allowed the beneficial ones to function. That is an oversimplification, but functionally accurate. His hepatic system does not merely filter foreign compounds. It evaluates them. It distinguishes between agents that threaten homeostasis and agents that support it. The threatening agents are dismantled with extraordinary speed and precision. The supportive agents are permitted to function, though at reduced efficacy and duration compared to their performance in other species. Dr. Vone, Judge Tessick leaned forward. You are describing a liver that makes decisions. Tessara looked up at her. This was the question she had been waiting for. The question she had been turning over in her mind since minute 19 of the first KR7 administration, when she had watched Cole Brannick's hepatic enzyme production spike in a pattern that no biological model could predict. I am describing a metabolic system that exhibits adaptive intelligence, Tessara said carefully. Not consciousness, not thought, but a form of molecular-level decision-making that operates faster than any cognitive process and with a specificity that our best engineered compounds cannot match. Mr. Brannick's liver does not simply process chemicals. It learns them. It categorizes them. It builds customized responses to each one and stores that response for future encounters. When we re-administered KR7 on day 6, 5 days after the initial exposure, his blood clearance time was 0.8 seconds. Less than 1 second. His liver had remembered the compound and had maintained its enzymatic architecture in a dormant state, ready to reactivate instantaneously. Molecular memory, Judge Tessick said. That is what I have termed it in my report, yes. His liver exhibits molecular memory, the ability to encounter a foreign compound, analyze its structure, build a targeted countermeasure, and store that countermeasure for rapid redeployment. No other species in our database demonstrates this capability. Not at this speed. Not at this specificity. Not at any speed, Tessara added and then corrected herself. That is, other species do exhibit hepatic adaptation. The Drelathi, as I mentioned, process 4% of KR7 on first pass. That percentage does increase with repeated exposure to perhaps 6 or 7% over a period of weeks. This is standard biological adaptation. What Mr. Brannick's biology demonstrates is not adaptation. It is something closer to. She stopped. The word she wanted to use was one she had debated with herself about for 3 days. It is something closer to engineering, she said. His body engineers its own defenses in real time. The gallery was silent. Tessara could feel 900 eyes on her. The judges, the spectators, the prosecution, the defense, the journalists. And Cole Brannick's, calm and patient in the witness enclosure, watching her describe the inside of his own body with the mild interest of a man hearing about the weather. Dr. Vone, Senior Advocate Torath stood. Let me bring you to the matter before this tribunal. The Conclave Bureau of Species Assessment has, based on your findings, proposed a reclassification of Homo sapiens from Threat Category 3, Standard Sentient, manageable with conventional protocols, to Threat Category 7. That is correct. Category 7 is, in the Conclave's classification system, the second highest designation. It is reserved for species whose biological capabilities render standard containment, sedation, and medical intervention protocols unreliable. There are currently 4 species at Category 7. You are proposing that humans become the 5th. I am recommending it, yes. On the basis of drug tolerance alone. Tessara looked at him. She had expected this question, and she had prepared for it with the careful precision that had defined her career. But the answer was not the one Torath wanted, and she knew it. Not on the basis of drug tolerance alone. On the basis of what drug tolerance implies. Explain. If a species' liver can analyze and neutralize 61 of 64 standard pharmacological compounds within minutes of first exposure, and can build permanent molecular countermeasures against each one, and can do this without any conscious involvement from the organism, the individual is not even aware it is happening, then that species cannot be sedated using standard protocols. It cannot be tranquilized. It cannot be chemically restrained. It cannot be rendered unconscious for medical procedures without developing compound-specific countermeasures that may activate during the procedure itself. It cannot be contained using any protocol that relies on pharmacological compliance. She paused. Every containment protocol in the conclave's standard manual relies on pharmacological compliance. Every one. The first line of every containment procedure for every species in the registry is, administer compound to achieve unconsciousness. For humans, that line is inoperative. There are physical restraints, Torath said. There are. This tribunal is not assessing physical restraint. It is assessing whether the current classification, which permits standard pharmacological protocols to be used in medical, containment, and security contexts involving humans, is adequate. My testimony is that it is not. Not because humans are aggressive. Not because they are hostile. Mr. Brannick has been entirely cooperative throughout this study. He has submitted to every test voluntarily. He has been polite, patient, and forthcoming. He is not a dangerous individual in the conventional sense. She looked at the witness enclosure. Brannick met her eyes. She thought she saw the corner of his mouth move, not quite a smile, but the architecture of one. He is dangerous in the pharmacological sense, Tessara continued, because his biology does not permit the tools we rely on to function. That is the threat that Category 7 addresses, not intent, but capability. Not hostility, but immunity. Dr. Vone, Judge Marath said from the far end of the bench, a Kalori military liaison whose presence on the tribunal reflected the security implications of the classification. I want to understand the practical consequences. A standard medical scenario. A human arrives at a conclave hospital with traumatic injuries requiring surgery. The surgical team needs to administer anesthesia. What happens? The anesthetic is metabolized before it can achieve sufficient depth for the procedure. The patient remains conscious. If the surgical team increases dosage, the liver upregulates and compensates. The patient continues to remain conscious. If the team introduces a secondary anesthetic from a different chemical family, the liver analyzes that compound independently and begins building a separate countermeasure while continuing to neutralize the first. How do you perform surgery on a human? Tessara looked at the witness enclosure. Brannick raised his hand slightly, as if asking permission. If I may, he said. Judge Marath looked at the bench. Korath nodded. You ask us, Brannick said. That is how you perform surgery on a human. You explain the procedure. You explain the pain management options that actually work for our biology, localized nerve blocks, regional anesthesia that targets specific nerve clusters rather than systemic metabolic pathways, or in some cases nothing at all. And you ask the patient to cooperate. Human surgery on Earth has always involved negotiating with our metabolism. Anesthesiology is one of the most complex specialties in human medicine, precisely because of what Dr. Vone is describing. Our anesthesiologists spend years learning how to work with the liver, not against it. They use combinations of drugs, precisely timed at specific concentrations, adjusted in real time based on the patient's individual metabolic rate. And even then, some patients metabolize anesthetics faster than expected. We call them rapid metabolizers. It is a recognized genetic variant, not a pathology. How common? Marath asked. Approximately 15 to 20% of the human population metabolizes standard anesthetics at an elevated rate. These patients require modified protocols. Some of them have undergone surgical procedures while partially conscious because the anesthetic was cleared faster than the surgical team anticipated. Partially conscious during surgery. It happens. It is not ideal, but it happens, and the patients survive it. Because the same biological system that is neutralizing the anesthetic is also managing pain response, stress hormones, cardiac function, and tissue repair simultaneously. The human body does not address one chemical challenge in isolation. It coordinates across every system at once. The tribunal absorbed this. Tessara watched Judge Marath's expression shift. The military liaison was recalculating something behind his eyes, and she knew what it was. He was thinking about containment facilities, about the holding cells on military stations where detainees were sedated for transport, about the emergency protocols that relied on chemical compliance as the first line of response. Torath stood again. Dr. Vone, I have one more line of questioning before I yield to the defense. Proceed. Your report mentions an incident on day 7 of the evaluation series. Compound number 19, the dermal absorption agent tessarine. Would you describe what occurred? Tessara had been waiting for this question. She had structured her report to make this question inevitable, because the tessarine incident was the moment her professional assessment had shifted from concern to certainty. Tessarine is a contact agent. It does not require injection. It is administered as a topical compound applied to the skin, where it absorbs through the epidermis and enters the bloodstream via dermal capillaries. It is commonly used in field sedation scenarios where intravenous access is not available or not practical. A standard dose applied to the forearm produces unconsciousness within 8 seconds in all tested species. Applied to Mr. Brannick's forearm. Applied to Mr. Brannick's forearm by Dr. Tessick, our dermal pharmacologist, at 900 on day 7. We applied the standard dose to a 10-centimeter patch of skin on his left forearm. The compound made contact with his epidermis at 904 seconds. What happened? His skin rejected it. The word landed in the chamber like a dropped instrument. His epidermis produced a lipid barrier, a thin layer of sebaceous secretion, at the application site within 1.7 seconds of contact. The barrier was chemically distinct from his normal skin oils. It was a targeted compound, generated on demand, that prevented the tessarine from penetrating the dermal layer. The compound sat on the surface of his skin without absorbing. Dr. Tessick wiped it away with a sterile cloth and examined the residue. The tessarine was chemically intact. It had not been metabolized. It had simply been denied entry. His skin produced a custom chemical barrier in under 2 seconds. In 1.7 seconds, without his knowledge, he did not feel it happen. He did not know it had happened until Dr. Tessick showed him the analysis. His response was, Tessara glanced at her notes. Huh. That's interesting. Then he asked whether the barrier compound could be synthesized for use as a general-purpose skin protectant. He was thinking about clinical applications before the data had finished loading on our screens. Dr. Vone, Torath said, the Bureau's Advanced Compounds Laboratory, has there been any progress in developing countermeasures? Dr. Mellish and his team at the Bureau's Advanced Compounds Laboratory have spent the past 17 days synthesizing novel agents, specifically targeting the CYPT enzyme family. Tailored compounds designed to inhibit the very metabolic machinery that makes humans resistant. They produced 11 candidates. And Mr. Brannick's liver neutralized all 11, the fastest in 6 seconds, the slowest in 2 minutes. And in the process of neutralizing the slowest one, a compound that Dr. Mellish described as the most sophisticated pharmacological weapon in the history of conclave chemistry, Mr. Brannick's liver identified the inhibition mechanism, analyzed it, and built a counterenzyme that not only destroyed the compound, but would also destroy any structurally similar compound in the future. His liver did not just solve the problem. It anticipated the next version of the problem and solved that too. She let the silence expand. 30 seconds, 40. That is why I am recommending Category 7, she said quietly. Not because humans are hostile, not because Mr. Brannick is dangerous. He is, as I said, cooperative and polite and genuinely interested in the science. He has been the best research subject I have ever worked with. But the question before this tribunal is not whether this particular human is a threat. The question is whether our infrastructure, our medical systems, our containment protocols, our security architecture, is equipped to handle a species whose biology treats our most sophisticated chemistry as a puzzle to be solved and discarded. My answer is no. The word fell into the chamber like a stone into still water. Tessara watched the ripples move outward, the shift in the gallery, the stiffening of the judges, the careful non-reaction of senior advocate Torath. Torath turned to the defense table. Your witness. Advocate Kelsareth rose. Her crest was still unsettled, a constant flutter of amber-tipped scales that Tessara recognized as the Veshan equivalent of sustained cognitive processing. The young advocate had been listening with the intensity of someone building something in her mind, and now she turned that intensity on Tessara. Dr. Vone, before I address your interpretive conclusions, I want to revisit the data from Day 3 of the evaluation. Compound number 12, the muscle paralytic vendrasal. Your report describes something your team called a countermanding signal. Would you explain? Tessara had not expected Sareth to go there first. It was a shrewd choice. The vendrasal data was the cleanest example of what she had been trying to describe. Vendrasal is a neuromuscular blocking agent. It interrupts signaling between motor neurons and muscle fibers, producing temporary paralysis. Standard onset is 6 seconds. In Mr. Brannick's case, the compound reached the neuromuscular junction as expected and began binding to acetylcholine receptors. The binding succeeded. His muscles did begin to lose function. For approximately 3 seconds, his grip strength dropped measurably. The paralysis was beginning. And then, at the 3-second mark, his peripheral nervous system produced a burst of acetylcholine, the neurotransmitter that vendrasal was blocking, at a concentration approximately 40 times his baseline. It was a deliberate, targeted neurochemical counterattack. His nervous system detected the blockade, identified its mechanism, and flooded the junction with enough of the natural transmitter to overwhelm the blocking agent by sheer volume. His grip strength returned in full within 2 seconds. By 8 seconds post-administration, his motor function was completely normal, and the vendrasal was being metabolized by his liver. His nervous system fought back. His nervous system identified a chemical blockade at the neuromuscular junction and independently produced a targeted counteragent in a concentration calibrated to overcome it. This was not a generalized stress response. It was not adrenaline. It was a precise, proportional, neurochemically specific countermeasure deployed at the exact site of the interference, in the exact quantity required, in under 3 seconds. Dr. Vone, you described Mr. Brannick's hepatic response as engineering, a liver that engineers its own defenses. You used the word intelligence, adaptive intelligence at the molecular level. These are remarkable claims. Are they yours or have they been peer-reviewed? My preliminary findings have been reviewed by the Kethara Institute's internal board and by 2 external reviewers from the Conclave Biological Sciences Directorate. All 3 reviews confirmed the data and the observational conclusions. The interpretive language, engineering, adaptive intelligence, is my own, and I stand by it as the most accurate available description. You stand by calling a liver intelligent. I stand by calling its behavior functionally equivalent to intelligence. I made the distinction in my testimony. I am not claiming that Mr. Brannick's liver is conscious. I am claiming that it demonstrates a capacity for analysis, response construction, and memory that exceeds any non-neurobiological process in our records. Sareth nodded. She had a way of holding silence that Tessara found unsettling, a deliberate pause that seemed less like hesitation and more like a hunter aligning a shot. Dr. Vone, did you ask Mr. Brannick about this? Could you be more specific? Did you ask the human about his own biology, about his awareness of these processes, about whether what you observed was unusual among his own kind? Tessara hesitated. This was the part of her testimony that she had not been certain how to present, because it was the part that had unsettled her more than any data point. Yes, I spoke with Mr. Brannock extensively after the evaluation series. He was remarkably forthcoming. He explained that. Perhaps Mr. Brannock should explain himself, Sareth said. She turned to the bench. Your Honors, I request that the subject be permitted to give direct testimony. Judge Korath looked at the other members of the tribunal. There was a brief exchange of glances, the kind that carried more information than words. The subject may speak, Korath said. In the witness enclosure, Cole Brannock uncrossed his ankles and sat forward slightly. He looked at Tessara with an expression that she had finally, after 4 days, learned to read correctly. It was not indifference. It was not contempt. It was the expression of a man who was genuinely curious to see how this would go. Dr. Brannock, Sareth said, you're a physician. Emergency medicine, Brannock said. His voice was calm and level, carrying easily in the chamber's acoustics. 12 years at Murren's Landing General. Before that, residency at Johannesburg Central on Earth. You're familiar with human biology. Professionally and personally. Dr. Vone has described your liver's response to these compounds as unprecedented, as a functionally intelligent. Is this news to you? Brannock was quiet for a moment. Tessara watched him compose his answer, and she recognized the process. A physician choosing words for a non-medical audience, translating complexity into clarity. The specific data is new to me, he said. Dr. Vone's team measured things that human medicine hasn't had the technology to measure at that resolution. The enzyme variants she identified, the CYPT proteins, we knew the family existed. We call them cytochrome P450 enzymes. We've been studying them for centuries. We knew they were highly adaptive. We knew they responded to foreign compounds with targeted metabolic activity. What we didn't know, or hadn't quantified, was the speed and specificity of that response at the molecular level. Dr. Vone's instruments are better than ours. But the underlying capability was known to you. Known and relied upon. Every human doctor trains with the understanding that the liver will attempt to metabolize any foreign compound introduced to the body. It's why drug dosing in human medicine is complicated. We have to account for first-pass metabolism, the percentage of a drug that the liver destroys before it reaches its target tissue. Different drugs, different rates. Different patients, different rates. We adjust doses constantly. We combine drugs with enzyme inhibitors to slow the liver down, so the medication can actually work. You have to fight your own liver to take medicine, Sareth said. Brannock's mouth did something that was definitely a smile this time. Slight, asymmetric, the expression of someone who appreciated a well-placed observation. That's a fair way to put it. Our pharmaceutical industry is essentially a centuries-long negotiation with our own metabolic system. We design drugs, the liver learns to destroy them, we redesign the drugs. It's an arms race. Has been for as long as we've practiced chemistry. And this is normal for humans, not exceptional, not a mutation, not a rare condition. Every human alive has this system. It varies in efficiency from person to person. Genetics, age, diet, health status all play a role. Some people metabolize drugs faster than others. But the fundamental architecture, the ability to detect, analyze, and build targeted responses to foreign compounds is universal. It's just how we're built. The gallery shifted. Tessara watched the tribunal judges process this. She could see the calculation moving behind their faces, the slow realization that what they had heard described as a remarkable individual capability was, in fact, a species-wide baseline. Dr. Brannock. Judge Tessick leaned forward, her silver scales catching the light. "You are telling this tribunal that every human on every world possesses the capability Dr. Vone has described. Every human's liver will destroy our pharmacological agents. With varying efficiency, but yes. Some faster, some slower, but the capability is innate. It's not trained, it's not optional, it activates without conscious involvement. Most humans don't even know it's happening. When you take a pill and it doesn't work as well as expected, you don't think my liver destroyed it. You think, I need a higher dose. It's invisible to us because it's so fundamental." "Invisible," Tessic repeated. "We breathe without thinking about it. Our hearts beat without conscious instruction. And our livers wage chemical warfare on every foreign compound that enters our bloodstream without any awareness on our part. It's autonomic. It's ancient. It's been doing this for longer than we've had language to describe it." Sareth stepped closer to the enclosure. "Dr. Brannick, you mentioned your experience at Murren's Landing. Have you personally encountered this kind of metabolic response in your patients?" "Every day for 12 years. The colony was on a world with aggressive native biochemistry, alkaloid-producing plants, mineral-rich water sources, airborne fungal compounds. Colonists arrived with no prior exposure to any of it. Within the first 6 months, every colonist's liver had built a comprehensive defense profile against the local chemistry. Children who were born on the colony showed even faster adaptation. Their mothers' immune systems had already cataloged the local compounds during pregnancy, and that information was transmitted to the fetus. Babies born on Murren's Landing arrived with hepatic enzyme profiles that included countermeasures for compounds their bodies had never directly encountered." "Inherited molecular memory," Judge Tessic said. "Epigenetic inheritance, we call it. The mother's body teaches the child's body what to expect. It's been documented in human biology for centuries. Pregnant women exposed to famine produce children with metabolisms that are more efficient at storing calories. Pregnant women exposed to pathogens produce children with immune systems primed for those pathogens. The liver does the same thing with chemical exposure. The mother's metabolic experience becomes part of the child's starting equipment. So the defense is not only adaptive in real time," Tessic said, "it is cumulative across generations. Going back to the very beginning, every generation of humans has faced a different chemical environment, different plants, different foods, different toxins in the water, different pathogens producing different metabolic byproducts. And every generation has added to the library. 4 billion years of chemical exposure, cataloged and passed forward. That is the system Dr. Vone's instruments measured. It is not a single individual's response. It is the accumulated chemical intelligence of an entire species' evolutionary history, compressed into the cells of one man's liver." The chamber held the kind of silence that follows an unexpected detonation, not shock, not alarm, but the suspended moment before the mind catches up with what it has just received. Tessara watched the tribunal absorb his words. She recognized the quality of the silence. It was the silence of a room full of intelligent beings arriving simultaneously at an implication they did not want to articulate. She articulated it for them. "If I may, Your Honors," Tessara said, stepping back to the lectern, "I would like to address the broader implication of Mr. Brannick's testimony, because it is central to my recommendation." "Proceed," Korath said. The conclave's pharmacological infrastructure was built on an assumption. The assumption is that biology is a solvable problem, that for any species, there exists a compound or combination of compounds that can reliably achieve sedation, unconsciousness, or metabolic control. This assumption has held for 912 species over 400 years. It is the foundation of our medical protocols, our containment procedures, our threat response frameworks, and our interspecies security architecture. She looked at the witness enclosure. Brannick was listening with what she had come to think of as his professional face, attentive, serious, the expression of a colleague at a conference. Humans invalidate that assumption. Not through resistance. Resistance implies a struggle, a contest that might be won with sufficient dosage or a more targeted compound. What Mr. Brannick's biology demonstrates is not resistance. It is metabolic sovereignty. His body does not resist our compounds. It deconstructs them. It learns them. It builds permanent countermeasures and stores them for instantaneous redeployment. And it does all of this faster than we can administer. She paused. We cannot outpace a system that accelerates in response to our efforts. Every dose we increase, the liver upregulates. Every new compound we introduce, the liver analyzes and catalogs. We are not fighting a static defense. We are engaging an adaptive system that improves with every encounter, that never forgets, and that operates at speeds our pharmacology was never designed to match. Then design new pharmacology, Toreth interjected from the prosecution table. Surely the Bureau's research division has been working on exactly that for 17 days. I discussed the results a moment ago. 11 novel agents, all neutralized. The human liver does not merely solve the current problem. It anticipates the methodology and pre-solves future iterations. Dr. Vone, Sareth rose again. One final question. Of course. You've spent 3 weeks studying Mr. Brannick. You've watched his biology do things that you, a researcher with 47 published papers and 6 years on the Cathara Institute's review board, describe as unprecedented. You've used words like engineering and intelligence and sovereignty. You've recommended the highest practical threat classification in the conclave system. Second highest, Category 8 is the maximum. Second highest, then. My question is this. In all your time studying him, all your conversations, all your observations, was there a moment when you were afraid of him? Tessara looked at the witness enclosure. Cole Brannick looked back at her. His expression had not changed, patient, calm, that mild curiosity that seemed to be his resting state. Behind him, through the transparent walls of the enclosure, she could see the prosecution table, the defense table, the curved walls of the chamber rising toward the tribunal bench, the 9 judges in their indigo robes, the gallery of 200 observers who had traveled from 6 systems to hear what she had to say about one man's liver. No, Tessara said. I was never afraid of him. That is not the correct framework. Fear requires a threat, an intention to harm, a capacity aimed at a target. Mr. Brannick has no such intention. He is a physician. He is a healer. He spent 12 years treating patients in a colonial emergency ward, and he volunteered for this study because he thought the data might help interspecies medicine. She adjusted her spectral lenses again. What I felt was not fear. It was the recognition that the tools I have spent my career building, the tools all of us have spent centuries building, the pharmacological infrastructure that underpins galactic civilization's ability to treat, sedate, contain, and classify biological organisms. Those tools do not work on this species. Not because the species is hostile, not because it is malicious, but because its biology was built in a chemical environment so hostile, so saturated with toxic compounds, that it evolved a metabolic system designed to destroy anything it does not recognize. And it does this without thinking, without trying, without even knowing. She looked at the tribunal. You asked if I was afraid. I was fascinated. I remain fascinated. I have spent my career studying how chemistry interacts with biology, and I have never encountered a system as elegant, as aggressive, or as utterly impervious to our interventions as the human liver. It is, without exaggeration, the most sophisticated metabolic engine in the galactic record. It took 400 years of conclave pharmacology and dismantled it in 41 minutes. But the fascination does not change the recommendation. The recommendation is based on fact, not feeling. The fact is this: we cannot sedate them. We cannot tranquilize them. We cannot chemically restrain them. We cannot assume that any pharmacological protocol will function as designed when applied to a human being. And every protocol in the conclave's operational framework assumes that pharmacological tools work. She closed her testimony file. They do not work on humans. The tribunal must decide what that means. The chamber held still. Tessara stepped back from the lectern. Senior Advocate Toreth sat down slowly. Advocate Sareth's crest finally settled, not into calm, Tessara thought, but into a kind of focused resolve. Judge Korath looked at the other 8 members of the tribunal. The exchange of glances was longer this time, laden with the weight of a species classification decision that would ripple through every medical facility, containment center, and security protocol in the conclave's jurisdiction. This tribunal will recess for deliberation, Korath said. Dr. Vone, you are excused. Mr. Brannock, you may remain in the enclosure or be escorted to the waiting area. Brannock stood. He stretched a simple, unhurried motion, rolling his shoulders and flexing his hands, and Tessara watched the tribunal judges watch him do it. She saw what they were thinking because she had thought it herself. Every movement he makes, every breath, every unconscious adjustment of posture, his body is running defensive chemistry at a level we cannot detect, cannot measure in real time, and cannot override. He is doing it right now. He has been doing it for the entire hearing. His liver is processing the trace compounds in the chamber's recycled air, the residual cleaning agents on the enclosure walls, the faint outgassing from the polymers in his chair. It is analyzing everything, remembering everything, building responses to everything. And he has no idea. Dr. Vone. Tessara turned. Brannock was standing at the transparent wall of the enclosure, looking at her. His voice carried through the acoustic ports with the clarity of a man accustomed to speaking in loud emergency wards. Yes, Mr. Brannock. That was a good presentation. Thorough. If you publish, I'd like to co-author the liver enzyme section. I have some notes from the Murren's Landing ER that might be useful. We had a run of alkaloid poisonings from native flora a few years back, and the recovery data correlates with your CYPT findings. Tessara stared at him. He was offering to help her document the very capability that had prompted the highest species reclassification hearing in 12 years. He was offering with the casual professionalism of one scientist, suggesting a collaboration. Yes, she said, I would appreciate that. Good, send me the preprint. I'll mark up the sections where I have field data. He smiled, a real smile this time, warm and unguarded and entirely human. And Dr. Vone, don't worry about the Category 7 thing. I understand the reasoning, it's sound. You're protecting your patients and your systems. That's what good medicine does. He turned and walked to the enclosure door, where 2 conclave security officers waited to escort him. The officers carried no chemical restraints. Tessara noted this, noted the absence of the standard sedation kit that accompanied every other subject escort in the building. They had already adapted to the data even before the tribunal ruled. The sedation kits would not work. Everyone in the building knew it now. The kits stayed in their cases. Tessara gathered her files and left the lectern. As she crossed the chamber floor toward the exit corridor, she passed the defense table. Advocate Sareth was organizing her datapads with the brisk efficiency of someone who had already begun preparing for the next phase. Dr. Vone, Sareth said without looking up, how many compounds are in the expanded pharmacological library? Beyond the standard 64. The full conclave library contains approximately 4,000 registered pharmacological agents. Sareth looked up, her crest flickered. Have you tested any of those? No, the standard 64 was the scope of my commission. But you will. Tessara paused. She felt the vertigo again, the sensation of standing at the edge of something much larger than a single study, much larger than a single hearing, much larger than the career she had built studying how chemistry talks to biology. Yes, she said, I will, and if the standard 64 is any indication, I expect the results will be consistent. Meaning? Meaning his liver will win. It will win every time, because it was built to win, not by design, not by engineering. By 4 billion years of chemical warfare on a planet where everything, the plants, the fungi, the bacteria, the water, the air, was trying to poison them. Their bodies are not defending against our compounds. They are defending against their own planet. Our compounds are simple by comparison. We are an afterthought. Sareth nodded slowly. She placed her last datapad in its case and looked at Tessara with an expression that was not adversarial, that had, in fact, never been adversarial. The young advocate had not been fighting the reclassification. She had been building a record, a complete, thorough, legally impeccable record of exactly what human biology could do. The defense had never been about Category 3 versus Category 7. It had been about ensuring that whatever the tribunal decided, the decision was based on the full picture. One more thing, Sareth said. Off the record, I spent 3 days preparing for this hearing by reading every pharmacological case in the Conclave's judiciary archives. 400 years of classification hearings, species assessments, threat level upgrades and downgrades. Do you know what I found? Tell me. In 400 years, every species that has been reclassified upward from a lower threat category to a higher one was reclassified because of something it chose to do, an act of aggression, a weapons development, a territorial expansion, a deliberate demonstration of harmful capability, every single one, until today. She closed her case. Mr. Brannick is being reclassified for something his body does without his knowledge or consent. He is being classified as a threat because of a biological process he cannot control, did not choose, and was not even aware of until your instruments showed it to him. That is unprecedented in the Conclave's legal history, and I wanted it on the record because I think it matters. Tessara looked at her. The young advocate's crest had settled at last, not into calm, but into something more like resolution. It does matter, Tessara said. She walked toward the exit corridor. The Presidium's main hall stretched beyond the doors, a broad, bright space of polished stone and diffused light. Brannick was already there. He was standing near the far wall, one shoulder against the stone, holding a cup of something that steamed faintly in the corridor's recycled air. The 2 security officers stood a respectful distance away. They were not watching him with suspicion. They were watching him with the particular attentiveness of people who had been briefed on his capabilities and were still processing what that briefing meant for their professional assumptions. Dr. Vone, he raised the cup in a small salute. Coffee, or what passes for it here. The Veshan delegates keep a brewer in the 3rd-floor reception room. It's not terrible. You found the Veshan coffee supply. I asked politely. The reception attendant was happy to share. She also asked me 14 questions about human caffeine metabolism. But that is an occupational hazard of being the most interesting liver in the building. Tessara almost laughed. The sound that came out was something closer to a controlled exhalation, the pressure release of a mind that had been holding itself in professional tension for 4 hours. Mr. Brannick, I want to ask you something that was not part of the formal testimony. Go ahead. On day 5 of the evaluation series, when we administered compound 41, the neurological depressant Calether, your brain produced a counter-compound that we had never seen before. Not in any species, not in any database. A neuropeptide that did not match any known biological molecule. Your brain invented a new chemical to defend itself. I remember the session. I had a brief headache, then I didn't. That neuropeptide, we analyzed it afterward. It was structurally elegant, more sophisticated than anything our pharmaceutical laboratories have synthesized. It was a perfect antagonist to Calether, tailored at the molecular level to neutralize its specific mechanism. Your brain designed it in approximately 6 seconds. Brannick sipped his coffee. And your question? Does it trouble you, knowing that your body is doing things, inventing things, that your conscious mind knows nothing about? He considered this. She watched him think, and she recognized the quality of the thinking, careful, unhurried, the deliberation of someone who took questions seriously. No, he said finally, it doesn't trouble me. My heart beats without my permission. My lungs breathe when I'm asleep. My immune system fights infections I'll never know I had. My liver apparently conducts molecular warfare on a scale that alarmed a room full of galactic scientists. None of it asks my opinion. None of it needs my opinion. That's not troubling. That's elegant. He looked at her over the rim of his cup. What troubles me is the gap between what my body knows and what my mind knows. Your instruments showed me things about myself that human medicine hasn't discovered yet. That neuropeptide, we don't know it exists. The CYPT enzyme variants, we haven't cataloged them. The dermal barrier response, we haven't described it in our literature. My body has been doing these things for my entire life, and I had no idea. That's not troubling either exactly, but it is humbling. Humbling. I'm a doctor. I've spent my career studying human biology, and it turns out human biology has capabilities that all of us have barely begun to understand. Your instruments are better than ours. Your pharmacological battery tested my body in ways that human medicine never has. And what you found was that we are more than we thought we were. That is a gift, Dr. Vone. Whatever the tribunal decides about Category 7, the data you collected is a gift to my species. Tessara adjusted her spectral lenses. She adjusted them because the alternative was to stand in a corridor of the Presidium Judiciary and feel something she had not expected to feel at the conclusion of a threat classification hearing. Gratitude. The human was grateful. He had been tested, measured, classified, and recommended for the second highest threat designation in the conclave system, and he was standing in a hallway drinking Vessian coffee and telling her that the data was a gift. She did not know what to do with that. I should file my report, she said. Of course, and I'll get you those Murren's landing notes. The alkaloid data is solid, 12 cases, full metabolic tracking. Dr. Tessick would find the generational transmission data interesting. The children born on the colony are showing hepatic enzyme profiles that include countermeasures for compounds their parents encountered before they were conceived. The library grows. He smiled again, that real, warm, entirely human smile. The library always grows. Tessara walked toward her office. Behind her in the corridor, Cole Brannick finished his coffee, handed the cup to the reception attendant, thanked her, and followed his escort back toward the waiting area. The security officers walked beside him at a comfortable distance. They carried no sedation kits. They carried nothing that relied on chemistry. The corridor was bright. The institutional light fell evenly across the polished stone, the embedded console panels, the slow-moving figures of judiciary staff going about their afternoon. It was the light of a civilization that had built its security on the certainty that biology could be managed with the right compound in the right dosage. That certainty had ended today. Not with violence, not with a demonstration of force, with a man sitting quietly in a chair while his liver dismantled 400 years of pharmacological infrastructure, and he asked for a glass of water. Tessara filed her report that evening. The header read, Pharmacological Assessment of Homo sapiens, Conclave Standard Battery, Full Results, with recommendation for immediate reclassification. The report was 412 pages. The conclusion was one sentence. Their bodies do not negotiate with chemistry, they dictate terms. The tribunal ruled 3 days later. Category 7, unanimous. The pharmacological protocols were withdrawn from human applicable contexts across 11 systems within the week. The sedation kits were recalled. New procedures were drafted, procedures that relied on verbal consent, physical accommodation, and cooperative frameworks rather than chemical compliance. Tessara published her findings 6 weeks later in the Conclave Journal of Xenobiological Sciences. The paper was the most downloaded article in the journal's 200-year history. Cole Brannock co-authored the liver enzyme section. His notes from the Murren's Landing alkaloid cases added 19 pages of field data that confirmed, with clinical precision, every observation Tessara had made in the evaluation suite. She did not tell him that the paper's appendix, section 14, for internal distribution only, contained a single recommendation that she had discussed with Judge Tessick privately in her chambers the evening before the ruling. The recommendation was this: begin testing the remaining 3,936 compounds in the expanded library. Do it quietly, do it thoroughly, and do not expect the results to be different. They were not different. They were never different. The liver always won.